Hi, class. Today we are going to talk about action potentials. So, you know that the nervous system sends and receives electrical signals through the form of impulses. So, we're going to see how that process starts with what is called an action potential. So, before an action potential or an impulse travels through a neuron, it is at rest. So, let's look at what happens at rest before it receives that signal. So, this is called resting membrane potential. So, the plasma membrane of cells in general, but we're going to focus on neurons today. The plasma membrane is polarized. We talked about this a little bit when we talked about muscles back in the muscle unit. But this means that cells are more negatively charged inside compared to their surroundings. And it's measurable. This is about negative 70 millivolts. And that polarization, that relative, negative interior in the cell, is caused by a disproportionate distribution of ions. So in cells, in neurons specifically, we're going to look at sodium and potassium. So more sodium is going to be outside the cell, more potassium is going to be inside the cell. But you might stop and think like, wait a minute, both of these things are positive, which is true. But because we have so much more sodium ions outside the cell, outside is relatively more positive compared to inside of your cell. What causes that uneven distribution of sodium and potassium in cells? Well, it's all about these sodium potassium pumps, and they are going all the time, not just in your neurons, but in your other cells as well. So for sodium potassium pumps, they take some energy, they're a sort of active transport, and they pump out 3 sodium ions for every 2 potassium ions that are pumped inside. And the result is the cell, again, is more positive outside and more negative inside. Sodium potassium pumps are just so common in your body that somewhere in maybe around 800 calories of what you eat a day just goes to powering these pumps. So these are kind of a big deal in your body. But let's talk about this situation, right? We're resting, your neuron is resting, something's going to come around, something's going to happen to cause it to send a signal. And that is called a stimulus. So a stimulus can cause the inside of a neuron to depolarize, right? Polarized means the cell's more negative inside. Depolarized means that cell's going to become more positive. And that's because sodium channels are going to open. So sodium's going to flood inside the cell. So some examples of stimuli include a neurotransmitter binding to a receptor. Right? We talked about acetylcholine and acetylcholine receptors in motor neurons, right? When we talked about how muscles contract. It could be a poke or a stretch. Think about like in your skin, something poking your skin, or think of an organ getting stretched out, or voltage change. And that's going to be a big one we're going to look at in the nervous system. So again, stimulus can cause the inside of a neuron to depolarize. And it's going to do that by opening sodium channels. Right? Sodium channels open. You can see this little diagram down at the bottom. Sodium floods in the cell, causing the inside of the cell to become more positive. And that changes the voltage across that cell membrane. So we were at negative 70, but now we have positive sodium ions flooding inside the cell, and that's going to make it more positive. And so if it reaches this point where it's about negative 55 millivolts, that's going to be the threshold to start an action potential. So we'll walk through this and put it in some diagrams and stuff in a little bit. Once you reach that threshold of about negative 55 millivolts, this is going to set off a series of rapid events in your nerve cell, your neuron, and it's called an action potential, which we also call an impulse, right? That electrical signal that gets sent through the nervous system. So that action potential, the cell is going to continue to depolarize. It's going to become more and more positive, and it's going to peak around 40 millivolts. You'll see some different numbers out there depending on the resource you're using. So we're just going to use a nice positive 40 millivolts because that's a really common number that you'll see. So once that cell reaches that peak, that positive 40 millivolt range, that is going to trigger something to happen. It's going to tell the cell to close the sodium channels, right? No more sodium is going to be flooding inside. And then it's going to open potassium channels and allow potassium to flow out of the cell. This is going to cause the cell to repolarize, to become more negative again, like it was when it started. All right, so it's becoming it's becoming repolarized, right? It's becoming negative again, but so many ions are flowing out of the cell that there's this little overshoot. So cells for a brief moment become hyperpolarized, right? We started at negative 70 millivolts. They are going to get down to negative 80. It's like an overshoot. It's too far. So too negative. But that overshoot triggers those potassium channels to close, and then it's going to be ready to return to resting membrane potential again. And so it's going to return to resting, that negative 70, and that neuron is going to be ready to be stimulated again and send another signal. So we are going to walk through all these steps in this diagram and kind of talk about what's happening with the different sodium channels, potassium channels, what's happening to the voltage of the cell at each point. So you'll see this on quizzes and tests in here. So make sure you know what is happening at each of these 7 parts. But actually, number 1 and number 7 are the same. So yeah, let's start with number 1. That's just your resting membrane potential. It's just waiting around, doing its normal cell stuff. That's going to be negative 70 millivolts. What's happening with our ion channels? Well, our sodium channels are closed and our potassium channels are closed. Just remember, in the background, which we're not really focusing on right now, you always have those sodium-potassium pumps going all the time. So we are going to look at what's kind of different for each of these steps. So sodium channels and potassium channels are closed. What does that mean for ion movement? Well, if these channels are closed, you can't get sodium or potassium going through the cell membrane. Right? So they're not flowing in or out of the cell. Stage 2, that is where we've had some sort of stimulus happen, which opens the sodium channels. So they're starting to open. And if we get enough of that sodium to come in, we'll reach that threshold of negative 55. So for step 2, we're going to say this is the threshold sort of level, negative 55 millivolts. The cell has become a little bit more positive because those sodium channels are opening, and so sodium ions are starting to flow into the cell. Potassium channels, potassium ions, we're still not seeing anything happening there. All right, so if it reaches that threshold of negative 55, then it's going to go to town. The action potential is going to happen. There's no stopping it. So step 3 we call depolarization. It's becoming rapidly depolarized, rapidly becoming more positive. Right? Membrane potential is rapidly increasing. We started at negative 70. We're working up toward that positive 40. What's going on with our ion channels? Well, our sodium channels are wide open. So we have sodium flowing into the cell, flooding inside, right, causing them to become more positive. Potassium, nothing's really happening there, right, until we reach that peak. So step 4 is that peak portion of our action potential, the highest voltage it's going to reach, and that's around positive 40 millivolts. That is going to be a big change. So we're going to switch over from sodium channels to potassium channels there. So for number 4, right, this peak, we get sodium channels are closing, so sodium stops flowing in. And then our potassium channels are opening, so potassium starts flowing out. Right? So it's sort of like a switch that gets turned on and turned off at that point. Right? And because of that switch, we're going to see now a rapid repolarization. Right? That membrane potential is rapidly decreasing, right, from this maximum amount of positive 40 down toward our resting potential again. So what's happening to cause that repolarization, right? Nothing with the sodium, right? Those are closed. Nothing's moving there. It's all about the potassium. So potassium channels are wide open. Potassium is just spilling out of the cell. And that's causing that cell to rapidly decrease in potential, right? Lower its voltage. Step 6 is that overshoot. We get that hyperpolarization where it gets even more negative than resting potential. And so that's going to max out at about negative 80 millivolts. Right? What's going on there? That's kind of the clue to shut it all down. So our sodium channels are still closed, but now the potassium channel is closing as well. So we don't get any movement of sodium and we don't get any net movement of potassium. Once those channels are closed, then we officially return to resting potential at negative 70 millivolts. Right? So no ion channels are going to be open. So sodium and potassium channels are both closed, and then we don't get a net movement of those ions either. No net movement of sodium or potassium. So you, again, will see this diagram again. It will not have the labels on the side. It will not have probably the words on here. So make sure that you know, both in like words and in numbers, for instance, what's going on at each of these parts of our action potential graph. I just kind of want to think about it big picture real quick. So this depolarization starts at one end of your axon, and that depolarization travels down it. That's what causes that impulse. So you get an area, not the whole axon, you get an area of the axon that's depolarized, and then that triggers depolarization in the next area, which triggers depolarization in the next section further down until that depolarization, that wave of depolarization, reaches the axon terminals at the very end. So it spreads like a wave. You get a wave of depolarization followed by a wave of repolarization, and then that wave of we're returning back to resting potential. And then one other quick thing is that some cells some neurons, some axons in your nervous system have myelin on them and some don't. So if you think about this bottom one, it's unmyelinated. That that depolarization wave has to go across every little portion of this entire axon all the way down. It's quick. It's still pretty quick, but it's not as fast as it is in myelinated cells. So when you have myelin wrapped around your axons, you can't have any sort of ion movement happening where there's those layers of myelin. So you can only have ion movement in these little nodes, these little spaces between myelin, those nodes of Ranvier. So that action potential is kind of neat. It has to jump from one section to the next, which makes it even faster. So myelinated cells just send that signal much, much faster because those ions only have to move through small portions of the cell membrane instead of along the entire length of the cell membrane. So just a reminder, we talked about nodes of Ranvier at the beginning of the unit. These little spaces, and that helps to function, especially the myelin, right? Myelin and nodes of Ranvier together help make these cells really quick at sending signals through your nervous system. All right, well, that is it about action potentials. Thank you.